Cellular robustness is not a byproduct of environmental flexibility. That's the topic I will be discussing in my first virtual online conference contribution. My name is Jürgen Zanghellini. I'm from the Department of Analytical Chemistry at the University of Vienna in Austria. So the question I'm gonna address in my presentation is which of these three strains is most robust. So basically we see our traditional lab strain, our E. coli strain, the MG 1655. Here we have a pathogenic E. coli, and in the back you see a Shigella strain. They share a large part of their genome. The question still though is there any difference in terms of robustness to these species? When we talk about robustness, we essentially first need to know what robustness actually is. And I've pulled out two definitions from the literature, and they define robustness as the ability to maintain performance in the face of perturbations and uncertainty. The Cellular definition from Computational is robustness is a property that allows a system to maintain its function against internal and external perturbations. So basically, when we are talking about robustness, we need to have three ingredients. First of all, we need to have a system. Otherwise, we can't talk about robustness because a single unit is not robust. It's just that single unit. We also need to have a function. That function is the function that is measured against the robustness, or that gives rise to the observed robustness. And we need to perturb our system in order to measure that in order to see if the system is robust or not. So how do we model these three ingredients? And for the course of this presentation, we're gonna look at genome scale metabolic models of microbes, and those are the systems we analyze. With respect to the function, we are looking at cellular growth, and cellular growth is the classical objective we use in flux balance analysis. And with respect to the perturbations, we look at loss of function mutations on the gene level or on the reaction level. So basically, what we do is we delete the reaction and see whether or not the organism is still able to grow. Okay, now we know that we know about robustness, and we can define it quite easily. But it's actually, despite this easy definition, it's actually quite hard to quantify robustness in a biological sense, or to have. And so the various measures have been put forward how we can actually quantify robustness, and they all differ in some respect, or they share some similarity as well. On the other hand, an engineer has no real trouble talking about robustness, and what they look from the engineer's perspective, they look at the reliability. So here is, for instance, a car, and we talk what's the reliability of this car. And when an engineer analyzes this question, he draws up, for instance, a reliability block diagram. And what we see here, this system or the car is made up of a couple of systems, just subsystems, that are the wheels, engine, transmissions, and we have two braking systems. And when is that car working? Essentially when there is a way from the start to the end. And so we find there's a way here and there's a way down there. So we basically find two ways that allow the system to work. And we basically can count the number of ways that get us from the start to the end. And this is the measure of reliability. A similar approach is, or viewed from a different angle, is the fault tree analysis. And here we look what happens when any of the involved subsystem breaks, for instance. So here we have the subsystems, the wheels, engine, transmissions, and the brake. And what happens if the wheel break we get the signal and the car is broken. The same for the engine and the transmission, but it's different for the brakes. So if the brake breaks, nothing happens, but if brake 1 and brake 2 break, we get the signal and the car is broken. Okay, so there is a very nice, so we see that there is a very nice and a lot of theory in the engineers world, and how can we translate that to the biological analysis? We are looking for something that works similar as in this reliability analysis. And in the reliability block work, we looked at pathways that get us from the start to the end. And in metabolism, there is actually a way to do this, and that leads us to the elementary flux mode analysis. Elementary flux mode is a unique pathway in a complete metabolic network that is in steady state, that is thermodynamic feasible, and adheres to all flux constraints that we apply. And basically, if we have all these elementary flux modes, we can characterize this whole metabolic network. So how does it work? So here's a tiny subware net, or here's a tiny metabolic network. Our flux is coming in, and here's our growth and metabolite A and B, and there are two pathways, two parallel pathways. So basically, we have two elementary modes, one this way and the other one this way. And this characterizes everything the cell can do. We can achieve the same characterization in a picture in the world of the minimal cut sets. And minimal cut sets are similar to the elementary modes, but we now look at ways that disable particular functions. So for instance, how can we suppress growth? Well, we could have a knockout here, then the organism doesn't grow. We could have a knockout here, and then it doesn't grow, or we could have two knockouts, one here and one there, and it doesn't grow either. And these three cut sets are called minimal cut sets, and they are also characterizing the complete network. And interestingly enough, the elementary flux modes and the minimal cut sets are two sides of one coin. If I have all minimal cut sets, I can calculate the elementary flux modes, or if I have all elementary flux modes, I can calculate the minimal cut sets. So the minimal cut sets are the ones we're gonna concentrate in the next couple of minutes. And that corresponds to the fault tree analysis from before. So basically, what we want to do is we want to calculate a probability of failure in metabolic network. And how do we calculate this? Well, we calculate the probability as the weighted average of the failure frequency. What's the failure frequency? Well, failure frequency is easy. We just calculate, or we just count the number of lethal combinations if we have D mutations or D deletions. And we calculate the ratio against all possible combinations with D mutations. If we have the frequency, the failure frequency for D mutation, we can calculate this for all possible Ds, and we weight this with a binomial distribution, and what we get is finally we get the expected value of the failure frequency. And that's what we call the probability of failure. Okay, now let's make this on this example. So basically we have here our simple example, and let's look at what happens if we do one knockout or one mutation. We could have a mutation here or there, and both of these lead to a lethal state. So we have two lethal combinations, and how many mutations are possible? We could have one here, one there, one here, and there. So we have four possible combinations or four possible mutations, and the ratio, the failure frequency is 1/2, weighted with the appropriate binomial coefficient. And doing this for all other possible combinations. So we have 2 deletions, 3 deletions, and 4 deletions, and we calculate finally the expected value of this failure frequency, and it turns out that in this example it's 0.20, so 20%. Whenever I do some mutation, or whenever mutations occur, there's a 20% chance that this will result in system failure or in the inability to grow. Okay, now how can we actually compute the probability of failure? It turns out that we can easily compute it if we have all minimal cut sets, and here's a formula. And what we need to do is we need to sum up all the minimal cut sets over the power set of all the minimal cut sets, and the power set means that we need to compute all combinations of the minimal cut sets. Now there's a couple of computational constraints and computational difficulties is coming to play now. Most of all, or most important of all, is can we actually compute all minimal cut sets? And the answer is no. And even if we could, then we have the problem to compute the power set of all these minimal cut sets if we just calculate, and that we can't do either. So there are some approximations that we need to do anyways. And we have been writing a paper that's currently in revision, and it turns out essentially we don't need to have all minimal cut sets. We just have to have those minimal cut sets with low cardinality. So that is the cardinality, the essential reactions. We have to have the synthetic pairs of lethal interactions and then triplets and so on. So the lowest cardinality minimal cut sets. And if we have this, we can calculate our probability of failure even in genome scale metabolic networks. And just let us test how it works. Here is E. coli. It's our standard E. coli genome scale model from Jeffrey Orth. And what we have been investigating, we have selected a minimal medium growing on glucose, and then we swap the glucose against all possible other single carbon sources. And what we find is the more complex the nutrients, the lower is the probability of failure. And that kind of makes sense. So if I have a complex nutrient that may have some carbon and also some nitrate in it, I'll essentially buffer the nitrate capabilities, and that allows us, that reduces the failure probability or makes the strain more robust. There are two observations we make, so two spikes. This spike here is the fatty acids. So down here are a short chain fatty acid, and here is a long chain fatty acid. And basically what we see is long chain fatty acid is more fragile than a short chain fatty acid. Why is this the case? That's actually a numerical or computational artifact, because we are not knocking out on a gene level, but on a reaction level. And if I'm feeding a long chain fatty acid, it just needs to be broken into a medium chain fatty acid and into a short chain fatty acid. So this is a linear pathway. Along this linear pathway, I have multiple reaction steps, and each of these could be a target of a failure or of a mutation, and that's why the failure probability is higher. We can similarly analyze this cluster, and it also ends up in some properties. But coming back to the question I started at the beginning, which of these three strains is now most robust? And it turns out it's actually E. coli. If I look at the pathogenic E. coli, that's a tiny bit less robust. And finally, the Shigella strain is noticeably fragile than the E. coli strain. We compared it against two other genome scale metabolic models, and these are two organisms, Pico and Hapti. These are two symbionts living in the stomach of cockroaches, and those are actually very fragile. So what is the difference between those five strains or these five organisms? It turns out one of the differences is the number of carbon sources they can utilize. So these symbionts here, they are living in a very constant environment already for millions of years, and they have a very particular requirement for their growth medium, while E. coli is a generalist that can grow on a very large range of different carbon sources. So this observation suggests that robustness is a byproduct of the environmental flexibility, and this is one of the classical theories that is around that it tries to explain the evolutionary origin of robustness. So can we actually test this? And what we did is we calculated the probability of failure for 500 different E. coli strains. So we found 500 different genome scale metabolic models, and for each of these, we calculated the probability and failure of failure. And what we see is E. coli, Shigella, and also Salmonella. They essentially sit on the same level. And there's not much difference, although they utilize between 120 and 180 different carbon sources. We don't see any trend in the probability of failure. The same is true in Trichoderma raceae. Also here, different Trichoderma raceae strains don't show any difference, or don't show show little differences in the probability of failure. What we do see is we see a huge or some substantial difference between Trichoderma raceae, which is more robust than the E. coli, but that's not an biological effect, but that's also a computational artifact. We looked into the biomass composition of E. coli, which is very detailed, while the biomass composition of the Trichoderma raceae models has only half of the biomass components of the E. coli strains, and that's why this strain is more robust, because it just has fewer components and therefore fewer chances of generating an error. Okay, so where does this idea come from that the robustness correlates with the number of carbon supporting environments or growth supporting environments? And the idea is that if you grow on multiple carbon sources, the metabolic network is tighter connected, but that is not true. That's not what we see here. Basically, what we have done here, we have compared metabolic network in a pairwise comparison, each network against another. And on the x-axis, you see a measure for the dissimilarity. So essentially, we see that all these E. coli networks are extremely similar, and that's why the difference in the probability of failure in the y-axis also doesn't change a lot. You can look at this from a different angle here. What you see here is the number of carbon sources. It grows on the x-axis, and the size of the metabolic reconstruction. And what you see is if you increase the number of, or if you increase the number of carbons you can utilize, you add on average 3 more reactions. And that are basically an uptake reaction, a transporter into the cytosol, and then one more reaction that connects it to the central carbon metabolism, but it doesn't lead to a tighter connection of the network. So where is actually the fragility sitting if it's not the carbon metabolism? And we have looked at all the minimal cut sets and analyzed where they are sitting in which subsystem. And what we find is the synthesis of the amino acid is the subsystem that is most fragile, and carbon metabolism is quite robust. So what you see here are the number of minimal cut sets in the biosynthesis of the amino acids in the blue one for different subsystems. And the blue bars are present in all of the 500 genome scale metabolic networks. And these white bars are the ones that are in some of the networks sitting. So basically what you see, amino acid is most fragile and carbon metabolism is actually very robust. That already brings me to my summary. I've introduced the probability of failure to you. I've tried to convince you that this is a consistent theory that's based on a rigorous probabilistic distribution, and it's calculatable in genome scale metabolic network. It decreases with the complexity of the nutrients, as we expect. We don't see any correlation with the number of growth supporting environments, as we could expect from one of the classic theories. At least that's true in E. coli. And we also find that the growth species comparison for Trichoderma and E. coli, for instance, is difficult because of differences in the reconstruction. We have calculated this all with some software that is already available on GitHub. There's a web interface that's coming soon. And there is already some publication available, and also some work that is to be published soon, hopefully. And that brings me to my conclusion, to my acknowledgement. I'd like to thank Julian, who was driving the work on the 500 different metabolic reconstructions of E. coli, and of my former postdoc Matthias and Christian, who were instrumental in the development of the initial theory and the software. And this work was partly in collaboration with Stefan Kamp from the MPI Magdeburg. And I'd like to thank you for your attention, and I'll hope you have some question which I'm happy to answer. My name is Jürgen Zanghellini, and I'm looking forward to hear your comments. Thanks.